go ahead and just help you all understand a little bit about our speaker today. Dr. Andrew Green is the springweed breeder and an assistant professor at North Dakota State University in Fargo, North Dakota. He began at NDSU in June of 2016. His primary focus at NDSU is variety development. Other research interests in his current role include early generation prediction of milling and baking quality, adult plant resistance to rust diseases, advanced techniques for fusarium head blight breeding, and experimental design and analysis for breeding. It is our great pleasure to introduce Dr. Andrew Green, and we thank you, Dr. Green, for joining us today for this webinar. Thank you very much. You are muted, so.
Here we go. There we go. Can everyone hear me now?
Yes.
Had to get that out of the way first. Well, thank you very much to Brian and to everyone at the NCI for inviting me to have this opportunity to come and speak with you today. And as Brian mentioned, there are some kind of distinct transitions in the presentation as I go through today. And if anyone has a question about anything before we move on, feel free to enter that and we'll try to take care of it. Otherwise, we'll have some time at the end. So as Brian mentioned, I'm the spring wheat breeder at North Dakota State University. And the motto that we've sort of come to adopt as I speak with our stakeholders and folks in the public is value on the farm and in the market. And I think that hopefully you'll see the fruits of our labor and the research emphases that we have kind of going to support that mission. So I'm just the fortunate person who gets to come and talk today, but there are a number of very talented individuals on our team, full-time research technicians, research scientists, graduate students, and a whole host of other undergraduate workers who also help make this whole thing go. Our program is fairly large and fairly intense in terms of field screening and the number of experiments that we maintain. And if it wasn't for this group, we wouldn't be accomplishing hardly anything. So today, I'm going to talk a little bit about our breeding program, but I think first it's kind of interesting and timely to discuss the impact of 2021, which is something on the forefront of everyone's minds as we look to the quality of the crop and we sort of review what happened and how we got here. Next, I'll discuss the overview of our breeding program so that everyone kind of gets a feel for how things move through the process in especially our early testing. We'll discuss some current research topics that we have in our program, things that we're currently emphasizing, and then we'll spend a bulk majority of the time in the second half of the presentation where I'll discuss a little bit about the quality parameters that we screen for and some approaches that we're taking to try to develop predictions for end-use milling and baking quality in a quicker fashion in our breeding program. So 2021 was a very difficult year. This picture was taken in Morton County, south of Mandan, North Dakota, in mid-July, and you can see that I do have a fairly big head, but my hat is not that big, and so you get a kind of sense for the context of just how short the wheat crop was in a lot of the state, driven primarily by the extreme dry conditions and also heat stress to a certain extent. But a lot of people will say, well, this must have been a very good year or an informative year to have experiments in the breeding program. And the question has come up, and it's a question we're still trying to answer. How do you select superior varieties during a historic drought? This is a case where we have screening all across the state, and our goal is to develop things that will succeed in production in North Dakota and the Northern Plains going forward. So dry weather is something that we'll have to navigate around. But when it's as bad as it was this year, how do we sort of work through that? So to give you an idea, if you're not familiar with the weather patterns from North Dakota for this summer, I've chosen to highlight four somewhat distinct locations across the state. And so you'll see here, there are separate charts for Carrington, North Dakota, Fargo, Minot, and Williston. And the top end, 100% on the y-axis here on the left, would be normal rainfall for that month. And so what you're seeing here are patterns where most of the locations spent the entire wheat growing season below the average monthly precipitation. This was coming off of a dry fall and coming out of a winter where there was very little snowfall, providing adequate moisture in a lot of these sites. And really, the uptick that we started to see in August was largely missed by the wheat crop. So the most critical months for growth and development of the upward yield potential in wheat are really happening in May and June. And if you look at the deficit, the amount below normal that we saw in these testing locations, it set ourselves up for some very challenging environments. And these data are all from the ENDON stations, which collect weather data, multiple parameters, but have a really nice tool for being able to look at these monthly precipitations. And so if we know we had dry conditions and we know that they were somewhat different across the state, but generally well below normal, was everything just a disaster? And the answer is no, not really. Drought is a very difficult thing to screen for in a breeding program because there's a lot of different places during the growing season we can experience drought. At planting, we might have problems with germination and emergence. If we have dry weather early on in the season, we might see establishment issues in small plot research. This can lead to inconsistent results. A lot of the times, if we're only trying to measure yield in this 50 or so square foot plot, having a good half of it, as you see in this picture here from Carrington this summer, emerge late or not at all, can give us a really difficult time in estimating the yield potential of these lines. So we might also have drought stress during other parts of vegetative growth, during reproduction of the plant when seeds are being set, and then during grain fill when we're really seeing those kernels get plump and full. And if we have drought stress during those phases, we might end up with low yields and poor results, but we might see consistent stands. So we might have a yield trial that's not as informative as it could be, but it was there and we were able to measure it and we were able to use and interpret the data for something. However, when we have drought during this entire phase of the life of the plant, what we end up with then is, what we would call a terminal drought. And in that situation, we have no results. So these are pictures in the bottom right. The no results is from Minot this year. There's about a stem every 8 to 10 inches. And most of those by this point that had anything growing on them had also been destroyed by grasshoppers. So this was a location where we didn't even measure yield. We stopped taking field notes shortly after head emergence, and we didn't get any data from that. So what did we end up with at the end of the year? These are pictures from the same experiment at 3 locations across the state, one in Castleton, North Dakota, here in Cass County in the east, one at Foreman, North Dakota in Sargent County down close to the South Dakota line, and then in Minot in north central North Dakota. And you can see the range of expectations we had here. An average yield in this experiment at Castleton, which ended up being very, very high. It wasn't because we had lots of rainfall in this area. It's because it received rainfall at the most critical times during the growing season. The average yield for that entire experiment was 101 bushels per acre, which is exceptional in any year, especially in a year like this. Down in Sargent County, we had uniform stands, as you can see. We had a really severe drought stress, very short plants, low yield expression around an average of 38 bushels per acre. And then you contrast that with the experiment in Minot, which was abandoned because there was essentially nothing there. So how does that happen? Well, the response varies tremendously, and you'll hear people say this field was just under the right cloud at the right time, and a lot of times those critical periods of growth in some locations, it varied. You drive down the road 2 miles and you might see a completely different story. And we also saw some real differences based on management approaches. So we're here in a lot of the state, we were raising crops on the reserved groundwater, which in a lot of places was not very extensive due to the less than productive winter in terms of snowfall. And so we saw differences based on row spacing, tillage, and especially previous crop. If we had a crop prior to our plots or our larger scale production fields, maybe following sugar beets or something that used up a lot more water, we saw less available water, lower overall yields. And so in the end of the day, we actually were able to salvage some information from this year's trials. So I think I've heard it said here in the last couple of weeks as we start to evaluate the crop, well, it was dry, there was almost no disease pressure. So what we probably saw then were high proteins, low yields, and a good quality crop. And I think that's probably going to be the case in a lot of the state. But to show you that there were differences in some of these locations, this is a plot of grain yield on this y-axis going from 85 bushels per acre up to 110, and grain protein on a seed basis from 11.5 to 16%. So what we see is this classic negative relationship between yield and protein, whereas protein goes up, yield goes down, and vice versa. You wouldn't expect to see this in a year where you had poor performing crops that were not using up that available nitrogen. And we actually, in this instance, which was here in Cass County, we saw a number of lines. In fact, most of the lines in this trial ended up harvested at a below 14% protein. So we were able to differentiate between these lines in terms of yield performance and for quality. And it'll be interesting going forward to see with no disease and the varying expression in protein level, how successful a lot of these trials were. But we did not have yield limiting conditions in all of our locations. And in fact, here we essentially under fertilized because we were expecting a lower overall yield than we realized. So that's kind of where we came out of 2021, looking at the overview of our breeding program in general to give you an idea of what it is that we're doing at these trials every year. As I said, value on the farm and in the market, that takes a lot of different faces here. Agronomic traits are the thing that we focus on first and foremost. We have to have a stable grain yield in order for a cultivar to be commercially competitive. It's our belief that genetic resistance to diseases, especially economically important diseases like Fusarium head blight, should be a priority. Where fungicide applications are not economical or not logistically possible, we do not want to release varieties in our program that are without resistance genes. And then sort of going through this in chronological order, if you will, through the growing season, the last thing on the list is not the least important, but it's the last thing that we measure in the sequence of the data for the year, and that's a strong end-use quality. So strong milling and baking for the processes that spring wheat is used for. And again, as we saw in that previous slide at Castleton, where we had that inverse relationship between grain yield and protein, it's always a little bit of a trade-off between end-use quality and yield performance, where we want to have the highest end-use quality possible while also maintaining competitive yields. So there's a few different ways that we do that. In terms of the list of things that we focus on, and when we're walking that balance or that teeter-totter, if you will, between end-use quality and yield, there are a number of things that I kind of identify as being unique to the desires for wheat farmers, for producers. Things like yield, straw strength to prevent lodging at harvest time, short-statured plants with a reduced plant height for less straw production, less tendency to fall over, and what I'll call harvestability, which would include the maturity and the ability to thresh these lines at the appropriate time, and also would include things like lodging. So those are important mostly only to the producer. The end users, however, have sort of a different set of needs. A high flour extract value, a strong dough with high water absorption, and a good consistent loaf volume for baked products. Those are things that a farmer would say are important to keep in mind, but these are specific desires that the millers and bakers would bring to the table that generally the farmer isn't going through in his checklist, his or her checklist. And then there are a few things in the middle that do actually overlap. Grain protein is necessary for that high end use quality, and also to prevent discounts at the elevator. A strong test weight to prevent getting a discount, which can be linked to that flour extract, but is mostly just important for grading. A strong falling number and good disease resistance, particularly for things like fusarium, which will have an impact on yield and end use quality. So what does that look like in the breeding program? The breeding program is this inverted triangle, if you will, where we go from having a large number of experimental individuals, a large number of genotypes that are screened at few locations, down to a very small number of individuals that are screened at multiple locations across the state. And so every year in the process, we are measuring these agronomic traits. We're screening the lines in disease nurseries. We're monitoring the end use quality, and we're eliminating individuals as we go each step of the way. And as we get further and further into the process, seed production begins for possible eventual release. A greater and greater number of testing locations starts to show up, and the number of traits that we focus on gets larger. So at the beginning, we might only be looking at protein, and that's sort of our estimation of the end use quality. By the time we get to the last stage in the process, we have over 27, well, there are 28, I believe, quality traits in our database that we have measured responses for on all the lines that make it to the end of this process. So this is something that gets more and more intense as we go. One of the key things about these early generations that's tricky is we have a high genetic variability. There are, within a family from a specific cross, there are a lot of individuals that may have the traits, the package of yield and disease and quality that we're looking for. But when there are over 3,000 of them in that first year, you've got to come up with some way to work through those and find the ones that you're after before you throw them away and sort of miss that opportunity. So we have a large number of lines and a small quantity of seed, which is key for the end use quality screening. So one of the things we've done, and I'm not going to belabor this point, in order to expedite this process and maximize our genetic gain, is we run our material through a number of winter nurseries in New Zealand, Puerto Rico, and Arizona. And the reason that we do this is to get multiple growing seasons in one calendar year, where we would be restricted if we only grew our experiments and increase these generations in North Dakota every spring and summer. It would add 3 to 4 years to the total process of how long we go from our cross to a released variety. And that entire process takes between 10 to 12 years if you sort of go about things a traditional way. And when we move things through these off-season nurseries, we can comfortably shave 3 to sometimes 4 years off of that process. So what else are we doing to try to push the envelope and accelerate that gain more quickly in the early stages of the breeding program while we've still got these large numbers of individuals? Well, one of the things that we're working through is an approach called genomic prediction, or if we utilize it in the breeding program, we would be using genomic selection. And essentially, what we're doing with genomic selection is taking complex traits that are usually controlled by multiple genes. So, for instance, we might look at a major gene for shortened plant height. The presence of this gene reduces the overall stature of the plant by, say, 6 inches. That's a single gene we can incorporate, we can track easily, and once it's there, it's there. Yield, Fusarium head blight, end-use quality, a lot of the things that really are deal breakers for us are controlled by multiple genes, and they have small effects, and they're very difficult to monitor and track. Some of these traits that we work with are also difficult to measure, or they're expensive. Our disease nurseries take a lot of time. It takes a lot of labor. The end-use quality traits that we measure, it's a lot of resources for the Wheat Quality Lab at NDSU and for the folks in our program to prepare the seeds, perform the tests, perform the analysis. Another problem with some of the things that we breed for is that the traits are not always expressed every year. So pre-harvest sprouting, for instance, in a year like 2021, we saw no pre-harvest sprouting in most of the state. We actually lost our Langdon location's grain because of pre-harvest sprout, but it just sort of was the icing on the cake that was the gift of 2021. We saw very little disease pressure. The problem is, in a year where we don't see those things, we still have to make advancements. That inverted triangles, we still have to cut down the number of individuals, whether we see those phenomena or not. So we have to have some way to make predictions for those and keep things moving. So what we do is we take, and this is not a unique approach that we've started. This is widely adopted in plant breeding. This is just a technology that we, in the last couple of years, have begun in our program, where we take marker data, molecular genetic markers on new lines, and we can predict these traits like sprouting or quality or yield or FHB resistance, which maybe haven't been measured before. And the way that we do that is hopefully outlined in this flow chart below, where we take this genetic data and then some trait data. So we have markers work, we have measured the trait that we're working with, sprout, quality, yield, FHB. We put those into a predictive model. And that model can get us a couple of different things. One, it can get us predictions for lines which we should be looking at releasing as varieties, and it can also help us select better breeding parents. We use that information to conduct more experiments to get more data, which goes back into the model and over time enhances the predictive abilities of our program's ability to estimate using only markers. So if we input this set of molecular markers and we know that they're associated essentially with these traits, what do we think the end use quality is going to be of this line, even though we haven't measured it in the field yet? And that's a technology that we're really pursuing aggressively. One of the problems with some of this work is, as we feed things back into that model with new data, we need good, robust scoring systems for all the traits that we work with. So visual scores work for breeding. On the left, you see fusarium damage. The one on the top looks very sound, no scab damage. The one in the middle is riddled with fusarium damaged kernels and probably has a high DON. On the bottom, it's kind of a mixture. If you were going to score it visually, I'm not sure what you would give it. We need quantitative measures for these things. Another example is the foliar leaf disease here, bacterial leaf streak. We know green plants are good and dead leaves are bad, but we need better information than that to plug into these predictive models to be able to advance the quality of our experimental lines. One of the ways we've started doing this year is through UAV images, drone photographs. We have a research scientist, Dr. Matthias, who's in our program, who's been training our research technicians and graduate students on collecting these data analyzing those for things like plot emergence, height, lodging, maturity, yield, and we're working with some diseases as well. So this is an example of, one of the aerial images that we took around harvest time. You can see each one of these little rectangles on the left is a yield plot. And in this picture, you can actually see some nice expression of lodging in some of the plants. This is the same picture, but zoomed in to give us an idea. We see varying stages of maturity here, different plant architecture. Some have a more dense canopy than others. What does that mean? Well, we take these images and we convert them into these spectral indices. In this case, green is indicating a full green canopy, which is full of leaves and plants, which are presumably photosynthesizing and creating higher yields, where red are more sparsely populated, less green, if you will. We might be seeing holes in these plots, or perhaps just low tillering expression. And so throughout the growing season, as we take these pictures and we correlate these data to the end yield, which we measure off of the combine, what we're trying to do is develop predictions for a lot of these important traits with some of these things that the eye can't see. So this is, again, not a new technology. This is not work that is unique to our program, but it's something that we've recently started doing to try to push things a little further. One of the other ways we started doing that is on the seeds once we get them home. Fusarium damaged kernels, as I'd mentioned, is a big deal. We've purchased this seed imager, which allows us to also measure things like test weight, seed size, kernel weight, and we're trying to train this to look for vitreous kernel percentage, for bacterial infection, other signs of stress. And so we can sort of have the images from the plots, images from the seeds, and then together, hopefully those give us an idea of the performance for these important traits. Pre-harvest sprouting, as I mentioned, is one of those inconsistent threats which we have to keep a close eye on. Many folks have memories of 2019 being a recent year where we had issues with pre-harvest sprouting across the state. And this is something that we have to measure and continually monitor, whether we see that in the field or not. Haley is a graduate student in our program, and she is working on developing new screening protocols and refining the ones we have. And she's also working on some genetic mapping to try to identify the genes that are in our program, which are responsible for resistance to pre-harvest sprout. So various methods, including a mist chamber with heads that are intact here for a screening that would more closely resemble what you see in the field, measuring seed dormancy on petri dishes in the lab. And the idea is this all goes back into those predictive models to be able to take a guess on sprout in a year where we don't see it in the field. So once we've gotten through all those things, if our experimental lines survive all those tests, one of the last things we start to really look at is the end use quality. And that has to start in those early phases of the breeding program. Our target goals for northern spring wheat in the public programs, which we've all kind of agreed to adopt, and so these are some targets that we adhere to in our program: 14.5% grain protein, strong test weight, 60 pounds per bushel. We want dark vitreous kernels, 80% or better is good. A strong falling number, so without that pre-harvest sprout. High flour extract, good water absorption, a stable dough that mixes well. A 10 to 15 minute phoenograph stability is the target. And then a good loaf volume as measured on the larger loaves or on the smaller ones that we sometimes monitor in the early stages of the breeding program. So the problem is there are probably many of you who are viewing this who are familiar with running a lot of these analyses, and knowing the phenograph traits and knowing the baking traits are critical. But when we go back to that triangle in the breeding program, the funnel, if you will, where there are over 3,000 individuals at the top, we don't want to throw some of those individuals away if they possess a good end use quality, but we can't do a phenograph on that many individuals. We can't bake those. So we have to find ways to predict the end use quality on small sample tests. Two ways that we've been working through that in the wheat quality lab here at NDSU are solvent retention capacity, estimating overall quality, gluten strength, and water absorption, and an instrument called the GlutoPeak, which is relatively new, a Brabender instrument, which is also, we're finding correlated with absorption, gluten aggregation, and overall quality when taken with the SRC and some of the other predictors in the program. We're still developing the predictions, but we think we're seeing things that tend to end up having better phenograph traits and baking traits at the end of the program. So when we get to that late stage, we have historically run mixographs. We are measuring these lines on the phenograph, and then we've got these little pup loaves to measure the low volume, the crumb, and the crust. And again, it would be nice if we could do this to each of these lines, but it's cost prohibitive. The timing is almost impossible. And so we have to find a way to guess at this. So the question then that I asked, and we're still trying to answer really, is what predicts a phenograph? How do we identify these lines while we still have them in the program in the top part of that funnel before we eliminate them? And I think what we're starting to settle on is that a combination of these genetic models using the marker work and genomic prediction, along with the chemistry models, the starch tests on the SRC and the GlutoPeak, some combination of those hopefully will get us more high quality lines as measured by the phenograph and in the oven. And so again, none of this is necessarily revolutionary, but essentially we have to figure this out before it's too late. And by the time we can afford to run a phenograph, we've already eliminated almost all the lines in the program. We've narrowed that down to a very select few. And being surprised at the last minute with those traits is not a good deal for anybody. So one of the last things I'll mention is about some of the work we're doing in hard white wheat in our program. You'll see a picture here, a seed sample of hard white wheat next to hard red on the right. And so the first objective might be painfully clear, but having a good white seed coat is our #1 objective. And I think as I show this second slide, you might see why that is difficult in a hard red spring wheat market to have that. And one of the reasons is that there's not much germplasm for hard white wheat that's adapted to our area. And when we have the high protein levels, the vitreous kernels that we are working with, they end up having this almost more amber color. They sometimes can be dark enough that they look like durum, to be quite honest. And so they don't sometimes have that bright white look that like a soft white wheat will. And so it's not always all that obvious. Our goal for this part of our program is balanced agronomic performance, aimed primarily at the West, strong end use quality. And this is in a market which would focus on whole grain milling here. And then genetic disease resistance is the same key that we saw with the red wheat program. But really, our #1 challenge with hard white spring wheat is pre-harvest sprouting. There's genetic linkage between the color alleles that make red wheat individuals more resistant to pre-harvest sprout than white ones are. So we're automatically fighting a battle there with the whites. One of the reasons, or one of the ways we're trying to work around that, I apologize. You can have the nicest phone in the world, but sometimes mute doesn't work. The southwestern North Dakota, somewhat arid environment. That would allow us to work around the pre-harvest sprout issue. So we want to get individuals that are resistant to sprout, but planting them in an area where they're less likely to see that stress is one of the ways to get around that. A lack of existing germplasm. There's really no easy way to do this. We're basically converting some of our high quality red wheats to having that white seed coat to try to bring along as much of the high quality red wheats that our program has been known for the last over 100 years and essentially make them white. Contamination is a big problem. You get red wheat into white wheat and it becomes a problem for everyone in the value chain. And so separate material handling equipment and facilities is the key there. And we've been very fortunate. We've got a good partnership with the Hettinger Research Extension Center. John Rickardson is an outstanding agronomist that we work very closely with at that center, which is kind of the hub of our white wheat activity. On the planter there, you'll see Adam Waltz, who's our research specialist working on white wheats. And then this lab in Van Ness Hall, which we've been able to set up specifically for processing the white wheats, helps us keep the seed stock of that program somewhat isolated from the red wheats to minimize that contamination. So the white wheats are a ways away yet, probably from being commercially deployed. The caveat on the last screen was that there's a very little market accessible to North Dakota farmers at this point for those products. The model and how all that would look is sort of being developed, but it's a thing that we hear feedback a lot about folks who are interested in these white wheats, and it's something that we're working on. So I think with that, that's a pretty good place for me to stop and see if we have questions or comments or complaints.
Thank you, Andrew. Excellent presentation. It's always so interesting to see how that works for those of us that are not involved with breeding. It's almost hard to really fathom and understand all that goes into this process. So thank you very much for that. So at this time, we'll go ahead and open up for questions. So if you have a question, please enter it in the chat box, chat function at the bottom of your screen. I had a couple of thoughts I guess, as people are maybe typing in there, had a couple of questions that came up. One is you mentioned, that you applying fertilizer, and you mentioned why you didn't apply the amount that you that you were expecting the yield. So that's a pretty important factor, trying to match expected yield with fertilizer. Any additional thoughts on that?
Commercially, it's a very, it can and should be very precise soil testing to fertilize for yield goals. But, when we applied fertilizer in the fall in the area of the plot that I showed there, I think we were probably fertilizing for around a 90 bushel yield. And as you saw with the results, we had some grain proteins that were on the low side, but we had some yields that exceeded 115, 120 bushel acre, on those plot estimates. So I think there's two things there. One is you're seeing some genotypes which just don't make protein. They're lazy. We've got some varieties in our marketplace that they don't fit the traditional spring wheat profile in terms of their protein content. So some of these are going to be down in that 12 to 13% no matter what you do. But the fact that the line had so many individuals that were close but still fell below the target, I think is telling us that we probably missed some of these in this area. And if we had fertilized for a higher yield goal, we might have gotten them. Now, how many years can you go into expecting to see yields this high without wasting money on fertilizer? That's a good question. I mean, costs are going to be up next year, and I'm not a soil fertility guy, but I think we did what we should have, but it's just an example of the extreme yield potential that we did manage to squeak out of a few of these places.
Great, great. Thank you. All right, got a question in from our viewers of the webinar today. Are you observing only modern varieties, or are you also monitoring heirloom or ancient varieties of wheat?
So we have had some historical trials in the last few years where we're looking at genetic improvement over time of varieties released through the North Dakota Experiment Station. We also do have a small trial that was planted at multiple locations across the country, actually, where we had some heirloom types under mostly organic production, which are being evaluated for a baking project on more of for the artisan market primarily. So there, I mean, we do get questions about that. We generally know about the profile of the historic varieties that we've had. Unless you get really lucky, to be completely honest with you, in a year like this where there was very little fusarium problem, the biggest limiting factor to those historical lines is a lack of scab resistance, so they can be very difficult to grow.
Very good. Thank you. All right. What are the reasons for the interest in hard white wheat?
What are the reasons?
Or what are the interests people have?
Whole grain products with that enhanced flavor profile that you can get with the white wheats being a little less bitter or a little more sweet, supposedly due to the tannins in the red seed coat. So that's what would drive that market. I know that the state mill does contract a small amount of that currently, which I think is primarily what that flour gets used for. But it's something that we get a lot of people that ask about whether or not we have anything that fits that profile for the whole grain market.
I know that in the past there's been some interest from some of our Asian markets for actually milling a refined flour just to make a whiter, cleaner product. So there is a little bit of interest overseas, but there again, getting that basically enough quantity in order to start driving those exports, it's always a challenge. So let's see here. I've got another question. Getting back to when you're making those crosses, okay, and you talk about genetic diversity and all these kinds of things. As a non-breeder, can you if you make multiple duplicate crosses of the same two parents, do you still get different results from those crosses?
Yeah, so I mean, generally in wheat, all of the lines we would be crossing with are fully inbred, so they're genetically stable, which is what we really strive for in seed production, so that when you plant seed of this variety this year, it's the same as it is next year. So the initial cross, that first generation after the cross is made, which we would call the F1 generation, that should be the same each time. But then when you self-pollinate that plant and you do the breeding process over again with different F2 populations, and if you selected those in different environments or for different traits, then you could end up with different results each time. So yeah, you could make you wouldn't have to make the same cross multiple times, but you could take seed from the same cross, and grow it in different locations and see how they're different once they were finished, and they would probably have diverged for traits of adaptation, depending on where they were bred. Does that make sense?
Yeah, it does. It's really interesting. Obviously, I'm not a geneticist, or a breeder, but I just think that's very fascinating how all that, and gene expression and some of that. It's just very, very interesting. And obviously, we don't have time to get into all of that information. We still have a few more minutes here from if anyone else has any questions to add. Got another one for you here while maybe people are typing. You showed that you go to Puerto Rico, New Zealand, and Arizona. Is there reasons that you go to one location for one stage and another one for a different stage? I just thought that's interesting that you choose these different locations.
Yeah, so sometimes we split them up across the three for risk management. For one, if we had everything in one place and something happened there, we would potentially lose, almost half the program all in one shot. So splitting them across locations, that's one reason. The timing is different for each one also. In the fall, New Zealand is usually the first to get shipped and planted, and then Arizona, and then Puerto Rico usually gets seeded last. And then when they're harvested, it's actually the other way around. It goes Puerto Rico, and then Yuma, then New Zealand, and then Arizona. So the time between harvest and when they need to be sent off varies, and then the time when we get them back varies. So that can affect the logistics of seed setup. So whether or not you have a lot to thresh before you get sent off, or when they come back, we don't want to send our biggest generation to Arizona, because we'll get those back in, say, mid-April. Well, we might be planting in the West, so we might send that material instead to Arizona or Puerto Rico and hopefully get a month shaved off the process.
Oh, thank you. That makes sense.
Yeah.
Okay, we have another question came in, and thanks to all of our participants here for sending in their questions. Are you doing any work on breeding for changing climate extremes?
In a way, that's kind of what we do every year, in the sense that we spread all the materials out across diverse locations and try to expose them to multiple conditions in a given year. So as I mentioned, we lost Minot due to heat and drought. Carrington was half and half. Langdon sprouted. Castleton, there was enough water. It was dry and hot. And Hettinger. And so because all the lines that will be released in 5 years are sort of a product of the most recent environment they were tested in and spread out across a very diverse state, that's kind of how we would prepare ourselves for lines that may face different conditions.
Thank you.
I do see Brian I just found the Q&A. There's a few more here. Besides the Buehler and the Quad, what's the largest mill you use before releasing a wheat variety? Some things are milled on a MEOG mill when they get to the Wheat Quality Council level. Generally, the larger scale samples are done on a Buehler. And then a follow-up, do you test the variety on a commercial mill? I'm not a cereal scientist, but they tell me that the Buehler is pretty predictive of the performance. So yeah, that's usually where we top out. When lines are screened in the Wheat Quality Council, though, they are milled commercially and screened for applications that vary based on the company. So we do get some of that data. And then Charlie, for my whole career, hybrid wheat has been 2 years away. Will it happen? How will this affect public breeding programs? So yeah, hybrid wheat is something that there are, to my knowledge, at least 3 private companies working on. I've been told with a fairly high level of confidence that they plan on releasing varieties quickly within that 2 to 5 year range. I think it probably will happen. I don't know at this point what would stop that, short of just the entire effort going away. I think there's always going to be a strong demand for pure line varieties, though, whether that's from a company or a university. And I think public universities are in a unique place to be relevant in hybrid wheat and pure line variety development, because parental development is obviously an important part of hybrid breeding, which is kind of what we're doing when we make pure line wheat varieties. And I think we're sort of trying to position ourselves to be relevant no matter which way the market goes. But I think there's going to be a strong demand for pure line varieties for a long time. People who like the ability to save seed, or like the reduced cost, even if they're in a certified seed only model, I would have to think it's still going to be cheaper to buy a pure line than a hybrid. So yeah, it's an economics question, but it'll be interesting to see. I mean, my career is a lot shorter than yours so far, Charlie, but I think your sentiment is probably the same thing I've seen, even just in the 10 years or so I've been around wheat.
So is it primarily just the economics of the seed cost, or is there some other reasons that's kept hybrid wheats from making it? Because I remember them talking about it in the 1980s.
Yeah, I mean, hybrid vigor is real. The response you get from a wheat hybrid is I mean, I've been able to witness that before. So you do get a yield gain. The problem is, yeah, the seed's very difficult to produce. It's expensive. And most of the time you're measuring like percent gains. So I don't know, say the state average of North Dakota might be, let's just say 45 bushels per acre if you average out the whole state. Well, even if you had a 30% yield advantage, which that would be pretty extraordinary, the amount of bushels that actually translates to relative to the seed cost might be where that whole thing kind of gets hung up.
Thank you. Another question here. I want to make sure this was from Rutger, so I want to make sure that we get his question answered. Are producers considering milling and baking characteristics and quality when making their planting decisions, and are they mainly focused on yield and disease resistance?
In my opinion, most or a higher percentage of North Dakota farmers, I think, consider this factor than maybe they do in other states or other regions, maybe would be more appropriate. I think that our spring wheat producers do recognize the value of wheat and the fact that it's largely driven by the end use quality. We're taking some steps in our variety development, or I mean our variety results guide and some of the publications that we release about quality measurements of our varieties in the variety trial to try to simplify that. So a more index-based formula where you can just see very quickly the relative end-use quality of all these lines when paired up, and say, go as high as you can on the yield or on the quality list while staying as high as you can on the yield column, and hope for the best. The sentiment that we don't get paid for quality, I mean, a lot of folks, try to beat that mentality back because it's really not true. The value of our products comes from the reputation of high milling and baking quality. And so without that, the market is sort of at risk of going away. And I think getting into the trenches on a yield battle is not the direction we want to go to maintain wheat acreage. So I think generally, there are some people who probably do just completely ignore it. But I think in general, the reputation of, for instance, some of these in the top left corner here, some of these yield kings that would be marginal winter wheat quality in the Central Plains, they always catch a little bit of acreage, but I think they get held in check by the fact that people pay attention to that quality. That's just me, though.
No, I think that's great, because I think it's really important that in order to maintain that premium market standing that we have, is to continue to educate producers and about the importance of choosing good varieties. So let's see, another question here: do private seed companies also focus enough on end use quality?
I think okay, so here's my disclaimer. I mean, I don't work for these companies. This is just on the outside looking in. Some do and some don't. Programs tend to have sort of a signature, if you will, in terms of the things that they focus on. And some of the private seed companies in our market that have a predominant share of the acreage do release varieties that are overall pretty high quality. Some, I think the case could be made by looking at the data, should put more emphasis on it. I think generally, though, the private seed companies that do put an appropriate amount of effort on it, you can tell that by checking the variety survey and seeing what producers are choosing. And I think that generally the trends over the last few years have reflected that the private companies that favor a more balanced approach do pretty well in the marketplace.
Thank you. No, excellent questions from our viewers today. So appreciate that. All right. Well, it's about 10:33 in Fargo. It looks like we've come to the end of our questions. I just want to thank, of course, Dr. Green for being our speaker today and providing an excellent presentation, and one that's been very interesting, just because of, obviously it's always interesting to see what's coming along in the breeding side of things, but with a drought year, and it wasn't too many years ago, we had terrible wet conditions, and so we're always dealing with issues here with weather being extremes. No matter what year, it's always extreme, seems to be. So thank you so much, and thanks to all of our participants today, and viewers of this webinar. If you have any additional questions, you can send them to to Dr. Green or to me. I know my email address is on the NDSU website, but it's brian.sorensen@ndsu.edu. So feel free to send those questions along. So on that note, Krista, I don't know if you had any final notes here for us, but I think we've come to the end of this webinar. So thank you all. Appreciate everything.